Hello, and welcome to day 3 of the Mitosis Notes. So we talked about the cell cycle, we talked about chromosomes, we talked about the introduction to mitosis, the interphase, we talked about all of that. Today we're going to focus on mitosis. So you want to look for the key words, and I'll explain it as we go. All right, so we have to start again with interphase, which is part of the cell cycle, the big part. And that was again, whoops, no pen. So that was again, let's go purple today. So that was G1, S, and G2. That makes up interphase. Now, what is interphase? It's when the normal cell is functioning, when you are, if you need to, replicating organelles. It is when you are making proteins. It's when you're doing anything a cell will normally do, including grow. It is the time after mitosis finishes just before the next part of when it comes around again. So remember, mitosis was just a small piece of the pie. So what's going on during interphase? Well, during interphase, you can see that thing called the nucleolus. That was in the first slide we did when we were looking at the cell itself. You can see the nucleus or the nuclear envelope. The chromosomes you cannot see because they are kind of all tangled up in that word which I call chromatin, which is where the DNA just kind of looks like that. So you can't see that on a regular microscope. And once again, for like the 20 millionth time, interphase is not part of mitosis. You know that's going to be a true and false question. So if it's interphase, once again, this is what you see. You can see the cell membrane right there. That's okay, that's fine. You can see the nucleus. You can see that kind of darker area right there. That's the nucleolus. And you cannot see the DNA. You can't see it because it's way too small for a regular microscope. But that's how you know this cell is in interphase, is doing normal cell things. Mitosis itself is divided up into 4 phases. You're going to see something called prophase, metaphase, anaphase, and telophase. And I'm going to teach you all of the steps in each one, and that will be today's video lesson. So let's start with prophase. Bunch of things are going to happen here, and most importantly is the chromosomes are going to condense. What that means is you are now going to be able to see them. If you remember from the last video, that's your chromatin. And then I drew your DNA kind of condensed like that. Once again, it's wrapped around the nucleosomes. I mean, it is a nucleosome. It's wrapped around the histones, and that's a chromosome. So you can see it. The centrioles are going to move to each pole. Once again, I'll show you what that means in the picture. You're going to have these things called the spindle apparatus, those microtubules we talked about at the end of the last video. They're going to start to assemble themselves. The nuclear envelope has got to go bye-bye because you have to be able to get to these chromosomes. So all this stuff is happening during prophase. And at this point, what the microtubules that have formed are starting to do is get the chromosomes organized. They're not at the center yet, but they're trying. So let's see what the picture looks like. You can see here that the chromosomes have condensed, so you can see them there. You can see that your centrioles, those are these green churro-looking things, they're starting to migrate to the poles. These green, like Spider-Man-looking things, those are your spindles. And if you look real close, you can see that the nuclear envelope is starting to break down. So those are your 4 things. Chromosomes condense, 1. Nuclear envelope breaks down, 2. Centrioles begin to move, 3. Spindle fibers form, 4. You know those 4 things? You cannot be fooled. That is prophase. Now this sometimes they're going to call prometaphase, but I'm not going to do that to you. I'm just going to kind of count this as prophase. But this here, and if you remember the last line of the slide before the picture, what it's starting to do is get these chromosomes organized. They're not in the middle yet, but they're getting there. And you can see that the nuclear envelope is gone. So I call this, I lump it all together in prophase, but if you take this in college again, they may talk about something called prometaphase, which is, again, in between prophase and metaphase. Now metaphase, now this is, again, the next big phase. The microtubules have pulled the chromosomes, so they are now aligned in the center of the cell, aka the middle of the cell. And that M and that M are how I remember that. And this is going to be called, when it comes up, the metaphase plate. And I'll just show you what it looks like in the picture. Metaphase plate, once again, is imaginary. Obviously, it's through the center of the cell. It's where the chromosomes all line up nice. And the spindle fibers, if all goes well, these spindle fibers have attached to the centromere. So if we kind of draw a little sister 1 and sister 2, the spindle fibers attach right there, and they come in over here. Now, if they, let's say, grab right there, or they miss, and they go for there, that's going to be a problem, because if they miss, they're not going to separate the sister chromatids correctly. So this is going to be I'm going to just write here. This is going to be a checkpoint right here, but we'll talk more about that in the last slide. But the checkpoint is how the cell knows, like, all right, oh, we made a mistake. But let's assume everything's good. So this here is your metaphase plate right there, where everybody's lined up in the middle. And if you notice, the spindle fibers, see them right there, that one and that one, they grabbed perfectly what they needed to. Everybody's good. Everyone's aligned nice. The chromosomes have attached. Everything is great. Life is good. Metaphase, check. Now, anaphase, this is where a lot of kids kind of get confused. Here, you've got rid of this cohesin protein. It's gone. Remember we said cohesins hold together the sisters? So that's gone. So now the sisters are free to separate from each other. And what's going to happen is this is supposed to be centromere, not centrosome. Centromere, sorry about that. Is going to move along the spindle fibers towards the pole. So what's really going to happen, it's kind of neat. I mentioned the word before, but kind of show you again. That region, that centromere region, has these things called kinetochores. And they are motor proteins. And what they do is they kind of, for lack of a better term, chomp and they move towards the pole. So it's pretty neat. It's the motor proteins in that kinetochore that move the sister chromatids, not the spindle fibers pulling like a rope. That's kind of neat. And now at this point, the chromosomes are going to move apart. They're now going to be called daughter chromosomes at this point. They have a new name. They went from sister to daughter because they're going to eventually be in their own cell. So here you can see they're separating. They are moving away from each other or apart from each other. And if you notice, that starts with an A, that starts with an A, and so does anaphase. And what's going to happen is, and it's in your notes, that this is going to ensure that both cells have the correct number of chromosomes. That's what anaphase does. If there's the wrong number, the cells will each die because they're not going to have the right genetic info. So there's that kinetochore we talked about, kind of chomping its way down, moving apart. Once again, it's still one cell. It's not two cells yet. Now in telophase, still one cell. What's going to happen here is now it's the exact opposite of prophase. The spindle fibers go away. The nuclear envelope is going to come back. And at this point, they're not each set of sister chromatids anymore. Now they're called daughter chromosomes. Daughter chromosomes. Now, they're in their own cell. They can begin to uncoil to turn back into chromatin. We remember, which is nice. So everything's good. And then the nucleolus is going to reappear. So what's really nice is we have now replicated the nucleus, but we're still technically in one cell. So it looks something like this. We'll talk more about it in the next video. But this is what we got. So there's your nucleus. There's that nucleolus region. There's your nucleus. There's your nucleolus region. The chromosomes have unwound, if you will, and the nuclear envelope is back. The nucleolus is back. The spindle fibers are gone, and that's your telophase. So again, this picture isn't great because it's kind of almost there, but you can see your nuclear envelope starting to come back. These guys have not unwound yet, but they will. This is early telophase. And then finally, if all goes well, the cells will split. And that is cytokinesis, the splitting of the cytoplasm. There's your nucleus, there's that nucleolus region, there's your nucleus, there's your nucleolus region, and the cell is ready to go. And the last thing for today, we'll pick up with this again on tomorrow's video. If you are a plant cell, and I'll describe it more tomorrow, you're going to have a cell plate. If you are an animal, you're going to have what is known as cleavage or the cleavage furrow. Once again, we'll pick up with that tomorrow. So I just wanted to get the basics down of mitosis and what happens and what the end result is. So hopefully that was helpful, and I'll see you next time.